As BCRs have a short cytoplasmic domain, so they cannot transduce signal to the nucleus for transcription and translation in response to antigen binding. Therefore, they require the help of accessory proteins. One of those accessory proteins is immunoglobulin alpha and beta dihydrodimer. This hydrodimer is also called CD79A and CD79B, A for alpha and B for beta. These are basically glycoproteins and are transmembrane, as you can see here. So these are the ones which are only expressed on the B cell. So CD78, 79A and CD79B are only expressed in B cells. So they have a single uh, immunoglobulin domain, which is extracellular, and intracellular. You can see they have motifs called ITAM, which stands for immune receptor tyrosine kinase based activator motifs. When these motifs are phosphorylated, they result in signal transduction. So students, it's time for our first concept checkpoint. You have to find out why only IgD and IgM can act as B cell receptor. The second question, you have to write down the role of CD32, surface molecule of B cells. Now let us study how B cell receptors are activated and signaling takes place. The first step in is the cross-linking of B cell receptors on B cell surface. The B cell receptors are present far apart on the B cell surface, but once they become activated by binding of antigen, they come close to each other. This process is called clustering. As you can see here, an antigen binds to the antigen, that is bacterial cell, binds to the B cell, resulting in cross-linking of the B cell receptors. This cross-linking then initiates signaling pathway. Clustering of the antigen receptors allows receptor-associated kinases to phosphorylate ITAM. ITAM binds to BLK, FYN and LYN and become phosphorylated, as you can see here. To this phosphorylated ITAM, SYK binds and results in signal transduction, which results in change in gene expression in the nucleus.
Antigen stimulation of the B-cell receptor is recognized as a critical mechanism contributing to lymphoid malignancies. Antigen binding induces the aggregation of the B-cell receptor with its co-receptors CD79A and CD79B. This leads to the recruitment of the tyrosine kinases LYN and SYK, which phosphorylate CD79A and B, amplifying the initial signal. SYK subsequently phosphorylates and thereby activates the lipid kinase PI3K, which in turn converts PIP2 to PIP3. PIP3 serves as a docking site for the cytoplasmic kinases Bruton's tyrosine kinase BTK and AKT. AKT activates the mTOR pathways, while BTK activates phospholipase C-gamma-2, which in turn generates a set of second messengers to activate protein kinase C-beta, which then phosphorylates IKK. Phosphorylation of I-kappa B by IKK results in the destruction of I-kappa B, leading to the release of NF-kappa B transcription factors that are then free to translocate into the nucleus, where they promote expression of genes regulating cell proliferation and survival.